This is a production of Cornell University Library.
Thank you. Well, I'm really happy that you could all come, and I'm happy to have a chance to tell you about the new book that we spent a lot of time working on. I guess I'll just start right away, and this is the pointer? Yeah, there we go, okay. So here is the book that this talk is going to be about, but let's go back a little bit, there we go, okay, to how this started. And it started with when I started on the faculty and I needed to design a course. And I designed a course called Natural Enemies, and it was about biological control, different aspects of biological control. And where was a textbook that I could use? I wanted to use a textbook. I needed an introductory book because it was like an introductory course. And here's what was out there. This one was too many stories. It was all case studies. This one was only theory, and this one had no illustrations. So my husband described this as the Three Bears meets the Little Red Hen, because I came out of that saying, okay, well, I better write my own book. Here's what I did. I wrote this book, and it was published in 2004. From when I signed the contract with Cambridge University Press until when it came out was 6 years. And so then when it was time to, there were a lot of changes in the field, a new book was needed, and so when it came time to do a second edition, I added another person to do it with. Remember the last one took 6 years? I thought it'd be faster to have another person. Well, it was 5 years. It's a little bit faster, but it's just a slow process. So, yeah. So, and this is the book that I'm going to be talking about. And in order to do a book, this book, I worked with a lot of people. Every chapter was the first edition was read by specialists, and the second edition was read by specialists, and asking people to use their illustrations. And for the second edition, one person who was really super helpful was Sana Gardescu, who's worked in my lab for a long time. She especially helped with all of the copyright permissions for figures. This sounds like something like nothing, but it actually takes an enormous amount of time and persistence. And then other folks that helped with it too, folks in my lab who helped with it, David and Carrie. So I wanted to say that it took a lot of time finding figures and then getting permissions to use the figures. And this is a picture, sometimes it was actually hard to get the figures. This is a picture of a little fish. How big do you think this fish is? Yeah, yeah, right, like this. It's like this. This is a statue in Sochi in Russia. I found out that it existed. I had to contact a colleague in Novosibirsk, who then contacted his colleagues in Sochi, who went to the place and went to the statue, took pictures, sent them to Novosibirsk, and those got sent to me. So, there's an inscription down here talking about how this little fish called Gambusia ate all of the mosquito larvae, which stopped the malaria epidemic that they were having in Sochi, and so they made this statue. I don't think there's very many biocontrol agents that there's a statue to them, and so I really wanted this picture. So sometimes it was hard to get pictures, just an example. So there's like 150 pictures. Some of them have numerous pictures in, and they're photographs and graphs and drawings. And here's, throughout the book, we also have stories, case studies. Every chapter has a couple of separate boxed stories that you can kind of read whenever you want. And so this is one that's about controlling, using a fungus to control kind of weed trees and bushes, and it's actually registered in Canada. So I'm not going to talk any more about that, but just to tell you, there's really diverse stories as you go through, and they're all stories that highlight different aspects of what's talked about in the text. So what does natural enemies mean? And I I thought I should actually talk about some actual natural enemies. And here I'm wanting to explain what is this insect and why is this insect on the cover of the book. It's called a green lacewing. And the adults actually eat pollen and nectar. That's why it's sitting there at the flower. And well, how is that a natural enemy? Well, it's the larval stage that's the natural enemy. They have these big, long, piercing fangs that they use to inject enzymes into whatever they've pierced. And then that liquefies it kills and liquefies the prey. And then they kind of drink them like a milkshake. So, oops, sorry. And so that's the larval stage. Here's the eggs. Because they're such voracious predators, the eggs, this is just what they do. The female creates the little stalks here and lays the egg at the end of the stalk. So then, otherwise, if all the eggs were right on the surface of the leaf, the first one to hatch would turn next door and eat all the other eggs, right? And so by having this kind of distribution, that's how they can avoid the first hatchling to eat all of the other hatchlings. Okay, so what is a natural enemy? The title of the book, organisms that kill, decrease the reproductive potential of, or otherwise reduce the numbers of another organism. And so this talk is going to be about using natural enemies for biological control. And there's been lots of controversy about what do you actually call biological control. And so this is the definition that we used in the book, that it has to be a living organism. A living organism that suppresses the population of some pest organism. So we wouldn't call GMO crops that are, you're just using the gene from maybe a bacteria. We wouldn't actually call that biological control. We'd call it something like biologically based control, but that's the general biological control community around the world pretty much accepts that. So we followed with that. Okay, so there are three main strategies for biological control. So how many here have kind of heard that biological control can be really bad? There's like a bad side to biological control. Good, okay. So I'm going to be talking about that. And I'm going to be talking about that because that's mostly relative to this first entry here, number 1, classical biocontrol. And really, biocontrol has these really diverse strategies. It's not only this bad one, but also the bad one, I think that we're solving some problems with it. Okay. Also, another aspect of biocontrol is that it's used in different ways. It's used, remember I just told you there's these different strategies, classical, augmentation, and conservation, and I'll be talking about those. But all of those strategies are used for biocontrol of insects. Biological control of weeds mostly uses classical biocontrol, and biological control of plant pathogens is mostly augmentation. And so by the end of my talk, you'll understand all of that terminology. We'll have a quiz. No, I'm kidding. Too much teaching. Okay, so I'm going to, in this talk, it would be way too much for me to try to cover everything, and so I'm only going to talk about biological control of insects. Okay, so classical biological control, that first category, that kind of category that people have heard is a bad idea. So what is it? And I kind of put the definition into kind of some steps of what gets done. So it's usually been aimed at invasive pests. And you figure out where the new pest came from, where the invasive pest came from. So like with emerald ash borer, which is just moving into this area and killing ash trees, that came from China. And so you go to the source where it's native, and you find the natural enemies of the pest, and then you bring them to the area that's been invaded. And then the old way to do this, the way this was traditionally done, was after kind of a minimal time in quarantine to make sure that you don't have any any contaminants or anything, then you release these natural enemies and they become permanently established and spread on their own. And so here are a couple of natural enemies that against the emerald ash borer. And then afterwards, we always say that we should study to see if the natural enemy actually provides control, how well it provides control. These are usually long programs, and frequently this last step isn't done as thoroughly as it should be. But these natural enemies I just showed you, those funny little insects that are just a couple millimeters long, I mean, they're really small. What are they? We call them parasitoids. Since it's a book on natural enemies, I figured I have to tell you some of these, kinds of natural enemies. These are used, it's the group that's used the most for classical biological control. And this is what they do. There's different parasitoids, and they do this in different ways, but they basically somehow inject an egg. Often this is into an aphid, and then you can see the larva growing inside of this aphid, and it's the parasitoid larva, and then here the host is dead and the new parasitoid is emerging. And it's like there's that movie Alien, and, and there's the alien comes out. Well, they stole that from parasitoid life cycles. So that's what happens with parasitoids. And you think, oh, that's so weird. What a weird life cycle. How could that be common? It's really common. It's actually done. There's lots of parasitoids. And here's one that's laying an egg into a fly pupa, and this one is used for control of flies on dairy farms in New York State. This one's laying an egg into a caterpillar egg. Here's one laying an egg into a caterpillar, and this one has a really long egg-laying structure we call ovipositor that it digs down into the wood and lays an egg on top of a wood-boring wood wasp larva inside of the wood. Finding it and laying an egg there, they're very successful. We have them emerging in barrels in our lab every year. So then another picture of a parasitoid. This is the first edition of my book. And this is a kind of parasitoid that they lay lots of eggs into a caterpillar. And then when they're ready to pupate, the eggs actually come out and spin up into this chrysalis kind of structure on the outside, cocoon on the outside, where they become pupae and adults. And then they chew their way out and fly off and find more caterpillars to lay eggs in. So pretty wild. But so this is the cover of the first edition. Okay, so parasitoids. Okay, so back to classical biocontrol and what is that? How does that work? And so, and why did it get such a bad rep? Well, so this kind of starts with in 1939 with DDT, and you all know that, DDT got used really liberally initially. And in 1962, Rachel Carson wrote Silent Spring, that was published, and the US EPA was actually started as a result of that. And the Silent Spring was actually started because of spraying DT against gypsy moth on Long Island. That was her really good examples that she had. So it's very close to entomology, but a lot of what she was saying, what was important about was impacts on bird populations and other populations, but people didn't really appreciate, I think, the entomology communities. But by the 1980s, I think the importance of biodiversity in entomology was really increasingly recognized. And so we were understanding that there was this big community of insects, and they were important and valuable. And so in that context, at the same time, the numbers of invasive species were increasing. Yeah, and you know that. But here's a kind of a recent graph that came out. Here's in 1990, the numbers of invasive species over time, and this is just increasing, and here we are up to 2000. And here's the number of classical biocontrol introductions. Why so much? Well, when there's a new invasive species, it's new, you don't know how to deal with it. One of the first ideas what to do is to spray insecticides. You spray insecticides or you do nothing. And the entomology community, basically their other fallback was to do classical biocontrol. That was the natural background of what you would do. And so that's what happened. Look at the number of classical biocontrol introductions. It kind of increased as the invasive species increased. And there's kind of a drop. This is probably World War I, and this is World War II. But up here, you're over 800. It's a lot. And this is what everybody's looking for. They were looking for the silver bullet. That back here at the start of all this in 1880, there had been some really silver bullet introductions that had super controlled pests really well. And so that's what everybody was looking for with these introductions. But here, how frequently is it successful? So the white bars are what you want to look at. And you can see that really about in here, it was like 10%. This is never really that great. Lots were being introduced. They might establish, but they weren't providing control, except sometimes. And they kept looking for the silver bullet, but really they were doing like a shotgun approach, right? And so as this interest in the biodiversity and the insect community started getting more recognized, the importance of it was recognized, then this started being voiced in the literature, that what were we doing releasing these natural enemies, and they were getting established, and sometimes we were seeing them attacking the natural community. And so was this a panacea, this classical biocontrol? Was it solving problems, or were we releasing Pandora's box that were going to be making issues? And so these are some papers between 96 and 2000. There were at least 50 papers about this in the literature. People just went crazy discussing this in their literature. Then there were some really heated words about this at scientific meetings. I remember that in an actual seminar that was given in Course in Mud about that time. So how risky is biocontrol, parasitoid drift after biocontrol introductions, re-examining Pandora's box. So there was a lot of controversy about this. And was this a bad thing to do? And so here's an example that this is a little fly. Doesn't look like much. It's a really little fly. But they lay eggs in caterpillars. And this is a gypsy moth. It was invasive, and that was what they were trying to attack with it. But this parasitoid is a generalist. A lot of parasitoids are really specialized in what they'll develop in. This one is a generalist. But it was released in 1900, 1910, and people weren't worrying then about the biodiversity, the native biodiversity. They were worried about making sure to not lose their crops, and especially orchard crops with gypsy moth. And so then what happened? Well, it turns out that it really likes to parasitize the native giant silk moths. And so now we think that one of the reasons the native giant silk moth populations are lower is because of the Compsoglyra that was introduced. But these decisions to introduce this were made in 1900, 1910. So it was a long time ago. So, okay. This was definitely happening, sometimes due to these we call this non-target effects, sometimes due to really old decisions that happened a long time ago. And then some people did surveys across biocontrol introductions against invasives just in recent years, in 2009, and they found 8 species out of more than 2,000 that caused population-level non-target effects. Everything else, it was just a really low-level occasional impact. but not actually driving down the population of the non-target. And so maybe it just wasn't studied enough. Maybe, you know, and the number 8 isn't very real, but this is all that we've actually found where this is having a population level effect. And so what's happened? Well, there's been a response, and so now the methods have changed, and now they do extensive non-target testing worldwide. This actually slows down the process a lot, but only specialist natural enemies are used, not generalist natural enemies, which, you know, before people didn't worry about that. And then they do risk analyses, and decisions about releases are made by diverse groups, the public and environmental groups, and the USDA and the EPA would all have a voice in looking at data, specificity data, in order to make decisions about releases. So then, is there a resulting slump in the use of classical biocontrol? While there's still more and more invasives, you know we're getting more invasives all the time. And you can see that here's 1980 to 1990s and 2000s. It really has decreased the amount of classical biocontrol going on. But is there still a reason to do it? And I would say yes, and I think the entomology community thinks yes. Because in some cases, when you have a really bad pest and there aren't any other options. So I'm going to talk for just a moment about emerald ash borer that's moving into our area. It's really a high priority pest. It's introduced from China, and in China, the ash trees that they have are resistant to emerald ash borer. In the U.S., that's not true. It kills our ash trees. And both in forests, you can see we have lots of ashes. Look at the ash, the amount of ash that's in New York. That's really higher densities than many other places. And here's an urban street. These are all ash trees in 2006 and 2009. It's really good at killing ash trees. If you drive through the Midwest and you drive along highways and you see lots of dead trees and you didn't remember that many dead trees before, those are probably ashes. So, so do we need classical biocontrol? And so this analysis was done looking at that there are 98 species of native ash-specific herbivores in North America. That will be extinct if there are no ash trees anymore. There's 12 species of ash. One of those that's native appears to be resistant, but the rest will be extinct, for the most part extinct, just like the chestnuts with chestnut blight. And so if this isn't enough to, you know, explain to you that you know, there is definitely a community associated with ash trees and the ash trees themselves. We actually use ash trees. The main kind of tree that's used for baseball bats, ash. So thinking about risk, to weigh one thing against the other, well, there is specificity testing, and the parasitoids that they're using sometimes will drift over and attack relatives of emerald ash borer, other closer related Agrylus species. That's the genus. That was just a pretty picture of this family of beetles. So I would say that the idea is it's more important to preserve the ash communities, and that's what is being done. And so after specificity tests, they're introducing 3 parasitoid species, and people have actively been working on that in New York State as well as in the Midwest, and there's one native parasitoid that's also attacking emerald ash borer. So just quickly, I wanted to say something about, I've spent a lot of my career working on gypsy moth and on Entomophaga maimaiga, a fungal pathogen that can also really control populations. And here's a smaller caterpillar, and the fungus kills the caterpillar, grows out through the skin, and shoots off spores. And those infect other individuals. Here, the bigger caterpillars, instead of pupating, they attach themselves to the tree trunks and die there. So in 1869, gypsy moth was introduced to North America. Entomophaga maimaiga, this pathogen, this fungal pathogen, was known from Japan. And in 1989, epizootics caused by this fungus occurred in 7 states, and then the fungus spread. We think it was an accidental introduction, but it just shows you that it's not only parasitoids that can actually control populations. I had to say something about this. I've worked on this a lot. Okay, so then I'm going to, I hope I've convinced you that that kind of bad biological control, there have been changes, and we're working on it, and hopefully it's safe for the environment. But another really active part of biological control is augmentation biological control. And so that's when controls achieved by natural enemies that are released where and when they're needed, and it's not for permanent control. And why do we need this? What about pesticides? That's what we use pesticides for, right? Well, there's the development of resistance, and just even this line here is the number of species of insects that are resistant to different pesticides. It's just increasing with a lot of pesticide use. That means we can't use the same pesticides all the time, right? This is a line that shows you how many pesticides are available. So this shows you how much pesticides have been being banned. You know, these were, this was kind of a large number here, and then the EPA started saying, hey, these aren't safe anymore, and banning them, a lot of these, from being used. So there's not as many pesticides to use, but companies make new pesticides all the time, right? The chemical industry, well, here is actually the trend in the number of new pesticides that are being developed all the time, and it's just decreasing. So there's a need. There's a need for something, and that's not only pesticides, not only chemical pesticides. And so that's augmentation biocontrol. It's also important for things like organic control. And there's a lot of reasons that people prefer this if it works. And this is a graph that shows you the number of predators and parasitoids, invertebrate natural enemies that are commercially available. In order to do augmentation biocontrol, there has to be an industry that's producing the natural enemies. And but look, there is. It's grown and grown. Here's 1960 and 2015. And by 2015, there's 400 different species that are available for biological control. So it's really a growing field, and people are using it a lot. John Sanderson, right here, who works in this field specifically, you can ask him about it afterwards for more specifics. And so this is just a couple of the big companies that actually send natural enemies all around the world. These are both in Europe. But then I wanted to show you this. This is a little company that they rear some of their own natural enemies, and then they also are a distributor for other natural enemies that they get from other companies. And this company is in Locke, New York. How many people know about IPM Labs? Yeah. So it's actually part of what we're talking about, this augmentation biocontrol, making these available. And I know that Carol provides a lot of parasitoids for the dairy industry for fly control, like I was saying before. And how can augmentation be so successful? Well, they've worked on quality control to make sure that the natural enemies delivered, remember they're alive usually. They have to be healthy and active, and you have to have a good way to release them. So they've also worked a lot on training growers so that the living natural enemies are released in the right way. So they get to, they persist in the crop and they get to where they need to be in the crop. These are used a lot in greenhouses, sometimes orchards, but there's also biopesticides, what we call biopesticides. And these are this term's in biological control, it's usually meant about microbes. There are other things that we call biopesticides, things that are like botanical pesticides, but I'm going to be talking about these that are biocontrol agents, living organisms. But do you like this term for using for a biological control agent? Who likes the term? Nobody. Well, we use this in the book to some extent because it's used in the literature quite a bit. And I think that it's used, why would it be used in the literature? Why would it be used, yeah? Yeah. And I think the idea is that growers might accept it more, you know, that, okay, well, this is something that something they could substitute for pesticides. So the microbes that are being used are virus and nematodes and fungus and bacteria. And there's hundreds of products worldwide that are based on these that are often for niche markets. They're for very specific situations, but sometimes they can be highly successful. So, and of course, this has been the most successful of any biological control agent in any kind of use is Bacillus thuringiensis. And it's a bacteria, and they attack caterpillars and different flies and different beetles. And actually, there's lots of other strains also now that they've found. But those are the main ones, the ones that are commercialized. And so this is one cell, and this is how it works, is all of them have a toxin crystal. And the crystals are different, and they have different specificities for what they'll kill. And this is actually, you know, it's this toxin crystal, the genes that create the toxin that get introduced into plants and that makes them resistant against caterpillar pests. That's GMO corn, GMO soybeans, well, no, not soybeans, but corn. So this toxin crystal, and this has been used extensively. Now it's used a lot in this GMO context, which we wouldn't call biological control strictly, but still organics and then things like the mosquito, they're used for mosquito control, Bacillus thuringiensis. Those are all, those are just sprays. They're available as that actual bacterial cells and not genetically modified plants. Okay, so this global biopesticide, if you read, you know, the kind of the economics market and the economics articles, the global biopesticide market is predicted to reach $7.7 billion by 2021, growing at a 5-year compound annual growth rate of 14.1%. This just, I'm not an economist, but this is just to show you that the number of biopesticides that are out there is increasing and increasing because there's need, right? Especially with all the organic crops and IPM and people wanting to use less chemical pesticides. So this market is growing. Okay, then the third way, I don't have that much to say about this, but this conservation biocontrol, it has nothing to do with releasing anything. It has to do with adapting the environment to increase the activity of the natural enemies that are already there, right? So this sounds really ecologically friendly, and it is, but getting it to work is the tricky part. So here, preserving natural enemies, that's one of the main strategies, and the main idea there often is don't spray pesticides when you're wanting parasitoids to be killing, you know, the pest in the field or in your greenhouse crop. And so this is based on reducing pesticides, but what I'm going to talk about here is enhancing natural enemy populations. Many people have wanted to get that to work, and lots of people have been studying this. It's been a very active area of research. And this is the whole idea, right? That this, if you have diverse communities around your fields, that the natural enemies will be able to increase and persist there, and then they'll move into the crop and will eat your pests. It sounds really good, and then it'd be a win-win situation by having this diversity around your crop. And so you can see here in a vineyard and then along other crop fields. And some recent papers have shown that it doesn't really always work. It's not, they did a big meta-analysis and it doesn't always work. And they're finding that they have to fine-tune it more in order to get it to work. But I'm going to tell you about a story where it does work. And so this is organic lettuce production in the Salinas Valley in California, big lettuce production area. And this is studied by Eric Brennan. And he worked on using flowers to, that's what he calls it, using flowers to replace insecticides in lettuce production. I really thought that sounded pretty good. And so here's a picture of his flowers. And so what's he want to control? This is romaine lettuce in Salinas. And they get these little aphids that get down in the middle of the romaine plants. This is organic. You can't spray it. You can't spray it. And even the things that you can spray, how can you get the things that you could spray down into the middle of a romaine head? And this is a romaine head that's cut in half, and you can see the aphids in there. And no one wants to buy their lettuce and bring it home and have the aphids crawl out. Yeah. And so then he found that a really good natural enemy that was around was this, and this is a hoverfly larva. And see, it's impaling an aphid there, and it's sucking it dry. So that's what they do. And they look kind of ugly, but they're doing their thing. And if the eggs are laid at the top of the lettuce plant, they will actually climb down inside and find the aphids and eat them all. So that's good. But the trick is you have to get them to the lettuce plants to have the aphids inside. How do you get them there? Well, you have to attract the flies that lay the eggs to the field. And how you do that is the flies eat pollen and nectar, and you plant flowers in the field, the right flowers, and then the flies will lay their eggs in the lettuce plants. And so this guy, Eric Brennan, spent a lot of years, and he actually figured out how much alyssum, that alyssum was a really good plant, how to plant it, how much he needed to plant. And he eventually came with a strategy that he figured out how to add alyssum plants to lettuce fields without loss in lettuce yields. So he kind of planted them at the edges of rows of lettuce. He didn't need any whole rows of alyssum. And this is going very well. But there aren't that many other examples in conservation biocontrol where you have such a really great success that's actually being used. So, but hopefully that will move forward because, like I said, there's a lot of interest in this field. Okay, so I'm just going to wrap up by talking about challenges to biological control. I hope I've convinced you that the bad biocontrol is actually kind of changed to, doing things a much better way and being used when it needs to be used. And the augmentation is this growing thing, but there's still challenges, right? There's going to be more invasive species. It's just, we were talking about that before, that there's lots of invasive species only going to increase. And the need for sustainability will increase as the human population increases. We've got climate change to deal with, and how that will impact biological control and pest populations. We're going to have to pay attention to that. I think it would be nice to increase use of biological control in public health. Sometimes some of the very worst chemical pesticides are used in kind of for controlling mosquito populations that need to be controlled. They need to be controlled, but hopefully we could do it in a safer way. And then we need to increase international cooperation with all of this. And then I think finally it would be nice to make sure that, that we have more acceptance that this bad rep for biocontrol gets corrected, and that people realize that there's an effort to make it environmentally safe, but we still control pests that have to be controlled, and show that biocontrol is used, when it's used, it's effective. Okay, so thank you.
With the microphone, and you can call on your questions.
Okay, good.
So was I understanding that augmentation means they're already there, they're doing something, and we're just putting more of them in, introducing ladybugs into my garden?
No, that's a good point. So lots of times now, well, the old view of augmentation, you could use something exotic and release it. And it might be something that you'd, so it'd be the same problem with this, is that environmentally safe? But now, a big emphasis is on finding natural enemies in that area, in that country, and mass producing those and making those available. So we won't be introducing new things.
So
Yeah.
the difference from classical is classical, you would go back to China to see what was there and bring that over. This one would look around and say, there's somebody here who might do this?
Well, or this one is whatever the industry is mass producing that would be available. Yeah.
But it's not,
And
you're not
for
introducing something invading from elsewhere.
No.
You're using something that's already in the neighborhood in some way.
Right. That's the attempt. That's what they're working on now. Also, with classical, the idea is that once you introduce it, you introduce some limited number. Maybe you only do one introduction, and then it's permanently established, and introduce it again. So that's different from, so it would have to persist somehow in the environment. With augmentation, you could, for your garden, you could buy them every year from the company that's mass producing them and release them. Then we wouldn't think that they would persist. So there's kind of
that you wouldn't expect them to persist.
Right, you would not.
Thank you. You mentioned gambusia fish right in the beginning. I guess they've been around a long time to control mosquitoes. How popular are they now and how successful?
Oh, that's a great question. People are not suggesting that you use them anymore because they're generalists, and there's a huge, push to not release generalists, and so they're generalists, and they will outcompete the other native species of the fish that are present. On the other hand, you always have to decide. So you have to decide. It's a risk analysis, just like I was talking about. And so you have to decide: is it more important to protect the natives? If what you're going to anyway. So, but it's not suggested to use them anymore. But I still think it's nice for Sochi to make a statue.
So going back to the fly larvae and the aphids, so you made the salient point that consumers don't want to go home and cut open their lettuce and find a bunch of aphids inside. How do you get the fly larvae to leave so that consumers don't take the lettuce home, cut it open, and find those there?
They don't take that long to develop, and I think that by the time they harvest the crop, they're gone. Those are gone.
Okay, so they kind of do their business and then move on quickly.
Right, yeah. But that's a good point. You don't want the flies emerging in your house either.
So, and the spotted lanternfly is the new scary invasive that's coming in, in Pennsylvania now, heading up to New York. So, what are the prospects for biological control of that one?
Spotted lanternfly. Does everybody know about spotted lanternfly? Yeah, it's a really big, it's like, it feeds on plants like a leafhopper, and it especially likes tree of heaven, which is like a weed tree, but so it's good that it likes a weed tree, you know, an introduced weed tree. On the other hand, it will eat a lot of other things, and it's been known to kill entire newly planted vineyards. So it's definitely a problem. My lab's been working on, we've made 10 trips down to Pennsylvania this year, and because it's just in Pennsylvania down by Philadelphia, but now they've found it in New York State, a couple of places, but in low populations. And we found 2 different fungal pathogens causing a lot of mortality in spotted lanternflies. So we're really excited about looking at fungal pathogens. Other than that, it's a group of, it belongs to a group of what we call plant hoppers. They really hop. When the little ones, you touch them, they're really strong hoppers, and there's no related insects in the Northeast. So there's there's little chance that there's going to be natural enemies that are used to eating something and attacking something in that family, because there's nothing else in that family that's in this area. So it's really a tropical insect, but appears to be surviving very, very well in the Northeast, which is too bad. So there's a lot of interest in getting to work on this thing, especially, you know, while it's still in a localized area, but it's increasing. The population's increasing and spreading.
So just a comment and a question. So Cactoblastis cactorum has a statue in Australia, the cactus moth, similar to the fish. And then my question is that between the publication of the first edition and the second edition, how did natural enemies and the use of natural enemies for biological control change, especially thinking about how genetic engineering has changed and then probably being used more frequently for pest management?
Well, so when it was first published, there was this, okay, so one big difference, we added a lot of new stories, new illustrations. One really big difference between the first and the second editions is the whole story about this non-target impacts. There was more information, and that had developed more, that we knew more about how bad is this, you know, non-target. Is there a chance that we should be using classical biocontrol, or maybe it should just not be used at all? So that developed, and we spent a lot more time talking about that. And then augmentation, we talked about how that's just been growing like mad. And so we talked about how and why that field has really increased. And then, am I answering your question? And then conservation.
I'm curious whether you saw any competition between biological control and genetic engineering and GMO crops.
Well, we decided not to actually cover GMOs. So we didn't actually talk about that at all. We talked about Bacillus thuringiensis as something that you'd spray, or something and it's used that way against mosquitoes. And we actually have one of our stories is about using an application, a formulation that's used in the Rhine Valley for controlling mosquitoes. So.
Are there certain kinds of organisms that seem to be more specific than generalists? So are funguses more specific, or are they equally likely to be generalists?
It depends on, there's different kinds of fungi, and it depends on the answer to that is, it depends on the kind of fungus. But then many predators are more generalists, and not all, but parasitoids, it seems like it's more of a general rule of parasitoids to be specialists. So, but that's all sussed out when we do non-target testing for, you know, to see how environmentally safe things are. Yeah, but there are trends. Any more questions? Wait.
I have a friend who has been for years part of the Ladybug Project here on campus. I'm sure you're aware of that.
The Lost Ladybug
Lost
Project.
Ladybug, yeah.
Yes.
So it's interesting to think about that we have there a species that is going extinct or could go extinct that serves a niche in you know, in our crops and in our ecosystems, that it could be a win-win if people can take that seriously and you know, use our natural, you know, insects and things that are failing and use them to, you know, I'm not explaining it well, but
No,
do you
does everybody
understand
know
what I'm
about
saying?
the Lost Ladybug Project? Have you heard of the Lost Ladybug Project? That there's a species of ladybug that is Coccinella novum notata. It was the state insect, and 9. And once an invasive ladybug got introduced, it was actually introduced for biological control, and it's a generalist, and we all think now that it was a very bad idea for it to have been introduced because it's such a generalist and it's so competitive and it's outcompeting the native species. And one of them that's being outcompeted is this novum notata. I think that the worry about this project was that it was absolutely extinct, and I think they're finding that it's not absolutely extinct, but it's been driven to low densities. So now I think that they're working on trying to grow them and release them again, and trying to see if there's some way to bolster the native populations. And so I think that's great. You're right. I mean, I think that that would be really valuable. Hopefully they have to find, though, a way to get around the invader ladybug, the Harmonia.
I think you mentioned that you had several fungal pathogens that might be effective on the spotted lanternfly. How do you come up with, do you have a drawer full of potential fungal pathogens? How do you come up with fungal pathogens to test?
So we're doing two different things. We've made 10 trips. This is in Berks County, Pennsylvania. We've made 10 trips there this season, and we've isolated fungi that we've found killing spotted lanternflies. So they naturally occur in the field. They're part of nature in Berks County, Pennsylvania. So that's part of what we're doing. The other part of what we're doing, there's a quarantine on campus. We've been working in there, and we've used a couple of different EPA-registered fungi that are known for being kind of generalists and for attacking hoppers, plant hoppers. And so we're just seeing whether those would be useful. Don't know yet. So they will definitely kill the spotted lanternflies. So that's good. But how to apply them, all that is a very early stage.
I was just going to mention the RSF collection at the USDA.
Oh,
I don't know
good.
what's going on with that, but I mean, are you collaborating with anyone?
They helped with doing the isolation of the harder fungus, so yeah.
As far as where you could select fungi from, that's,
Yeah.
yeah. You
But-
might explain what that collection is.
Yes, so there's a fungal culture collection. It's a giant container full of liquid nitrogen that they've stashed different fungal isolates of fungi that kill insects from around the world. And I think they have like 14,000 isolates now. So it's an enormous resource. And it's just up Tower Road. And I've worked really closely with these folks for years. They're USDA Agricultural Research Service. And so there's a great, huge resource of different fungal isolates to test there, or to examine. But in order to use something actually for control and get it out there so that the organic vineyard people don't have all of their grapevines killed by spotted lanternflies, these things have to be registered with the EPA. And in order to register a new fungal pathogen, let's say fungal pathogen with the EPA, that can take $1 million for all of the testing and lawyers and also something like 4 to 5 years. So it makes sense to test the ones that are already EPA registered first before going to the culture collection, because that you better have a lot of resources to use that.
So I was just wondering, I know you mentioned that there's a bacteria that produces like crystal toxins or something.
Yeah.
So I was just wondering is there any other examples of like genetic engineering of things like bacteria or fungi, like to specifically like address the pathogen, but like, I don't know if like the EPA would like allow that, but is there like examples of things like that, or even like genetic engineering of like a natural insect in order to be able to like metabolize, I guess, like other pests that they normally wouldn't eat, something like that?
Yeah, people have worked on that to some extent with different pathogens, seeing if they can insert something that causes virulence in one kind of pathogen into another, in order to improve, make it so it kills the host more quickly. And so there has been some playing around. Nothing is a product, but there has definitely been some investigation along those lines.
I just had a general question of what is the process your lab takes in like assessing the risk of introducing species?
So that's actually, it is often done by universities, but it's also done a lot by USDA. And so I've never, well, maybe a long time ago when I was in grad school, but, and then people weren't looking at the risks. So, so my lab doesn't do this. But I know what the procedure from colleagues that work in the USDA, and they would have, it's a long process of coming up with what species should be tested in order to do this non-target testing. Close relatives a little further away still on being related to the actual pest in order to see how generalist is the natural enemy. And then that has to all be approved. The list of things to test has to be approved by this whole board that I told you, of all the people that would decide whether the non-target testing results showed that it was safe to be released. So.
Well, thank you, Anne, for a really nice talk, and thank you all for coming.
Thank you.
This has been a production of Cornell University Library.